Hi, I'm delighted to give a presentation about the compelling advantages of incorporating sulcal width as a biomarker for local atrophy in the aging brain, supported by an extensive multi-sample study. Cortical thickness is the go-to feature used by researchers trying to assess quantitatively local atrophy, while radiologists prefer using sulcal width, but only as a means to qualitatively assess cerebral atrophy on MRIs. Sulcal width, sulcal aperture, and sulcal opening are all synonyms for the same thing, the mean distance between the two walls of a sulcus. We had some intuitions about the advantages of considering sulcal width rather than cortical thickness, although no comparative study of both features exists yet in the literature. Indeed, the evaluation of cortical thickness relies on gray and white matter segmentation. Yet the blurring between gray matter and white matter junction increases with age on T1 weighted MRIs. It should be much easier to measure sulcal width, which relies on the more contrasted segmentation between gray matter in gray and the CSF in black. Sulcal opening or sulcal width should also yield better signal-to-noise ratio, as it incorporates the atrophy of two cortical ribbons. On top of that, it is possible that sulcal openings may unveil some other aging process which is only present in the subcortical structure and not in the cortical thickness. For example, in the third image, we have the FLIR MRI of a patient with extensive white matter hyperintensities, which are present in some gyri and appears only on FLIR MRI and not on T1 weighted MRI, but it may influence the sulcal width of the neighboring sulci, but not the cortical thickness. In order to examine these two features, we use two atlases. The BrainVisa atlas, which enables the computation of both cortical thickness and sulcal opening across 62 regions of interest per hemisphere, and the desiccant atlas, which provides exclusively cortical thickness measurements across 34 regions of interest per hemisphere. Presently, the desiccant atlas is widely employed to investigate brain atrophy. We assessed comparatively the performance of sulcal opening and cortical thickness using the BrainVisa atlas, as well as evaluated its performance in relation to cortical thickness obtained from FreeSurfer on the desiccant atlas. To tackle this issue, we leveraged two large cohorts with distinct characteristics. UK Biobank, comprising 20,000 subjects with available MRI scans, age, and gender information. Since there are no specific inclusion criteria, UK Biobank is commonly regarded as a representative sample of the general population. In contrast, Memento exclusively recruited subjects presenting isolated subjective cognitive complaints or mild cognitive impairments while not demented. Besides, during 4 years of follow-up, 208 subjects out of the total 2,000 and something subjects were then diagnosed with dementia. To conduct our analysis, we extracted the two phenotypes, the cortical thickness and the sulcal opening, from T1 weighted MRI scans of all participants using the aforementioned atlases. Throughout the whole study, we separated our analysis by sex. Upon examination of the sulcal width separately by sex in the subjects from UK Biobank, the general population cohort, an interesting observation emerged. There is an acceleration in the local atrophy rate among women for many sulci. For instance, in the case of the right superior temporal sulcus, we first notice an initial phase characterized by a plateau, followed by a change in slope. Here, we suggest modeling the progression of the sulcal width, or the cortical thickness, as a piecewise linear function to capture the sudden acceleration of atrophy at a specific point, visually observed in several sulci. Rather than considering all data points for each sulcus, we fitted these two segments using the 90th percentile line to focus on at-risk subjects in the UK Biobank dataset to filter out outliers and account for intersubject variability. The final parameters were determined through bootstrapping to avoid overfitting and compute confidence intervals. In essence, we generated 100 samples with replacement from the original UK Biobank dataset, trained segmented linear models for each sample, and recorded the parameter values to compute the final estimates and confidence intervals. Shown here is the final model for the right superior temporal sulcus, along with the variability of the 90th percentile line during bootstrapping. With this segmented linear modeling approach, we can effectively quantify the age of acceleration, representing the inflection point at which sulcal width begins to increase more rapidly, or the inflection point at which cortical thickness begins to decrease more rapidly. This feature enables convenient comparison of different sulci based on changes in slope and age of acceleration, providing valuable insights into the relevance of the initial null hypothesis, which states that sulcal width experiences a sudden acceleration at a certain age. If the null hypothesis we initially assumed holds true for a specific sulcus, then the stability of the age of acceleration during bootstrapping, despite resample variability, should indicate a pronounced and easily detectable point of acceleration. This leads to a smaller confidence interval for the age of acceleration, or as we suggest calling it, the tipping point age. Conversely, if the change in slope is not sufficiently pronounced, meaning that the initial and final slopes are very close, even slight fluctuations in the resamples can result in significant variations in the age of acceleration, leading to a higher variability of the tipping point age during bootstrapping. In other words, a significant variation in the calculated breakpoints during bootstrapping, as depicted by the red line on the slide, suggests a less distinct change in slope and questions the relevance of the null hypothesis. Therefore, with the tipping point age, we get a feature that quantifies the degree of acceleration in the atrophy of any given neuroanatomical feature. In this brain mapping, which displays the standard deviation of the age of acceleration during bootstrapping for each sulcus, the ones shown in blue indicate a stable tipping point age, suggesting a well-defined and easily identifiable inflection point. With the concept of the tipping point age, we can compare the performance of sulcal width with the two other cortical thickness measures. To select the sulci that truly exhibit acceleration in atrophy, we apply the following two criteria. The change in slope must occur in the correct direction, namely a higher final slope compared to the initial slope for sulcal width, or a lower final slope compared to the initial slope for cortical thickness, indicating accelerated thinning of the cortex. Additionally, the age of acceleration must remain relatively stable during bootstrapping. Therefore, for visualization purposes, we have set an arbitrary threshold of 3 years. The question is, how many sulci actually exhibit acceleration in atrophy? For the sulcal width measured by brain visa, there are 58 sulci showing acceleration in women, which are here classified by increasing confidence interval for the tipping point age. In contrast, there are only 14 sulci showing acceleration in men, indicating a clear difference between men and women. However, for the cortical thickness measured by brain visa, there are only 4 sulci showing acceleration in both men and women, and they are not exactly the same. This suggests that cortical thickness measured by brain visa is not able to detect acceleration in atrophy, whether because the feature itself is not sensitive to the process, or because the data is too noisy. Furthermore, for these sulci, the age of acceleration are much later compared to the corresponding sulcal width, suggesting that sulcal width captures neurodegeneration at an earlier stage. When we look at the cortical thickness measured by FreeSurfer on the desiccant atlas, the modeling provides more results. There are more regions of interest that exhibit distinctive acceleration in atrophy based on cortical thickness. However, this acceleration still occurs much later than in the sulcal width, as we can observe, for example, in the right superior temporal region. On average, the ages of acceleration are 57 years for sulcal width and 70 years for cortical thicknesses measured on the desiccant atlas. Sulcal opening seems to reflect earlier neurodegenerative processes than cortical thickness. What we would like to know now is the meaning behind this acceleration of atrophy in the trajectory of sulcal width. What process does it translate? From now on, we will focus exclusively on sulcal width and its models. We start with an interesting side result. The trajectory differs significantly between men and women, and once again, this difference was only detected in sulcal width, not in cortical thickness. Men seem to be subject to accelerated atrophy much earlier than women. Their initial and final slopes, computed on the age range of UK Biobank, are very similar, as opposed to women. To investigate the process behind these atrophy accelerations, now let's focus on our second cohort, memento, which consists of subjects at risk of dementia. We started by correlating the sulcal width of each sulcus with a free recall memory test, and we obtained this graph. By ranking the sulci in ascending confidence intervals, meaning that the sulci that show a pronounced acceleration in atrophy are on the left, we observed an increasing trend in correlation with memory tests, represented by a correlation coefficient of 0.47, which is quite substantial. This correlation, we will call it the double correlation coefficient. This means that the sulci most associated with memory test scores are those that exhibit a pronounced acceleration of atrophy during aging. Thus, by simply selecting the 90th percentile line from UK Biobank, we have discovered that physio-pathological processes in general cohorts can be reflected in an at-risk cohort using a very simple model based on sulcal width. If we fit this piecewise linear modeling to all quantiles, not just the 90th quantile, we obtain these plots for all the regions of interest. Regrettably, the tipping point ages for every quantile do not seem to follow a well-defined trend, the best-case scenario being that tipping point ages decreases for higher quantiles. However, it is noteworthy that there is still evidence of atrophy acceleration even in the healthiest subjects, namely the lowest quantiles. This observation indicates a transition towards a general phase that is characteristic of aging, whether it is considered normal or pathological. We've been talking about the lower quantiles as the healthiest subjects in the general population and the higher quantiles as the most at-risk subjects. But can we prove it? For each quantile, we calculated the double correlation coefficient between the length of the bootstrapping confidence interval of the tipping point age and various cognitive tests. The correlation reaches its maximum for the highest percentiles, consistently across all cognitive tests except for phonemic fluency. This suggests that we have successfully targeted the highest risk subjects within the general cohort using only sulcal measures, which indicates the potential of incorporating sulcal width into normative models to be utilized in clinical settings. Furthermore, we observe that correlations with cognitive scores are largely similar, indicating that this phenomenon is not necessarily specific to memory or the classical form of Alzheimer's disease. It appears to match impairment across multiple cognitive systems. In conclusion, sulcal opening is a feature that is extremely interesting and too often overlooked to study neurodegeneration from a purely neuroanatomical point of view, and we suggest that incorporating it more systematically in studies on the aging brain would open up new avenues for research. Thank you for your attention.